Hello, college biology. Welcome to part 6, phase 4 of cellular respiration. This would be the electron transport chain. Quick review. In glycolysis, we broke glucose down into pyruvate, and we made a couple electron carriers. In the transition, we took pyruvate into the mitochondria, we changed it to acetyl-CoA, and we made a couple electron carriers. In the citric acid cycle, we did a big loop of chemical reactions, starting with citrate and acetyl-CoA, and we made a ton of electron carriers. So now we've built all these electron carriers during this whole part as we've been breaking apart glucose. It's time to use them in the electron transport chain. That's phase 4. And so if you look at this, you can see that this blow-up is a blow-up of the electron transport chain, and you can see that it's occurring right here in the inner membrane space. Well, we can kind of zoom in and see that, okay, there's in the membrane, there's a bunch of transport proteins, there's a lot of stuff going on. Let's take a closer look. So this is our electron transport chain, and it involves a few things. One, let's figure out where we are. This dark red area, this is the mitochondrial matrix, the innermost section of the mitochondria. This is our inner mitochondrial membrane. This is our inner membrane space, and this is our outer membrane. And so this would be the cytoplasm. This stuff is outside of the cell. So back inward we go. We have been making all of these electron carriers, and finally there's a place to take those electron carriers, something to do with all these electrons we've gathered from glucose, and that is these guys right here. These are transport proteins that pump hydrogen ions out into the inner membrane space. Now, hydrogen ion is the same thing as a proton, and we're going to talk about why that's significant in a minute. Before we get there, how does this work? Well, electrons are polar. They are negatively charged. We know that polarity can affect the shape of proteins. Well, we drop off an electron, it changes its shape, it isomerizes, that results in its ability to take a hydrogen ion out here and put it out here. Now, once that happens, the electron's not done. There are the other electron carrier molecules that accept the electron, and they kind of shuttle it to other proteins. So with one electron, we can get this pump to work, but we can also get this pump to work, and then other carriers will take it over here, and we can get this pump to work. And finally, what happens to that electron? Well, we've got some oxygen over here. We've got some extra protons floating around. We take the electron, we take oxygen, we take some extra protons, we make water. So we breathe in the oxygen we breathe in to accept all the extra electrons we have broken away from glucose, and then we make some water, which is useful for the cell anyway. So that just stays put. So we've done it. We have taken the electrons, we've triggered all these pumps, we've used our electron carriers, we've got hydrogen out into the inner membrane space, we found a way to deal with that extra electron we've been carrying, we've put it into water, yay! So what? Well, you're right, now we have just a bunch of hydrogen in the inner membrane space. What does that matter? Well, we did before say that hydrogens are the same thing as protons, and that is this now is referred to the proton motive force. These high concentrations of protons are called the proton motive force. Now, what good is that? Well, we know that particles move from an area of high concentration to an area of low concentration. And when they move across a membrane from an area of high concentration to a low concentration, with the help of a transport protein, that would be facilitated diffusion. Well, we are going to use that tonight. We are going to see that these high concentration of hydrogen ions are going to pass through a special dual action transport protein. Not only does this protein help hydrogens go from the high concentration to the low concentration, it also is an enzyme that triggers the change of ADP and phosphorus into ATP. So every time a hydrogen passes through this, ATP gets made. And since we have been using the electron carriers to trigger these pumps, to pump a ton of hydrogen out there, we got a ton of hydrogen coming in through facilitated diffusion. So ATP, ATP, ATP. Every single time that hydrogen comes in, we get ATP. This miraculous dual action protein is called ATP synthase. It's a protein that builds ATP. That's what that name means. So let's summarize. The electron transport chain occurs in the inner membrane. It's got a lot of membrane-embedded proteins, a lot of this fluid mosaic stuff going on. It's between the mitochondrial matrix and the inner membrane space. The electron carriers that we've been charging with electrons through this whole process finally deliver their electrons to hydrogen pumps, triggering their transportation of hydrogen ions from the matrix to the inner membrane space. Once we have all those hydrogen ions in the inner membrane space, ATP synthase uses the hydrogen concentration gradient. So there's facilitated diffusion from the inner membrane space to the matrix of hydrogen ions to trigger isomerization so it can produce ATP from ADP and phosphate. I hope this helps. Struggle well.